Hello friends, let us learn about the other virus, which is Hepatitis D virus. So in Hepatitis D virus, it is a member of the genus Delta virus genus. Excuse me. And it is a defective RNA. So it cannot infect alone. So this Hepatitis D virus always co-infects with Hepatitis B virus, or it needs a helper virus, that is Hepatitis B virus, for its replication. Okay, so this hepatitis, it is slightly smaller than Hepatitis B virus. It's slightly smaller than Hepatitis B virus, and it is also formalin sensitive like Hepatitis A virus, and it is 35 to 37 nanometers virus with hybrid structure. It is a nucleocapsid, and it expresses delta antigen. Okay, so this delta antigen is completely different from the other types of Hepatitis B antigens. The genome of the Hepatitis D, it is small and 1700 nucleotide, circular and single strand RNA of negative polarity. It is an RNA virus, and it is circular, and it is single stranded RNA, and it has 1700 nucleotide. Okay, now Hepatitis D viral RNA, it contains many internal complementarity, okay, and this can also fold on itself by internal base pairing. Just by internal base pairing, it can fold on itself, and this forms an unusual, very stable rod-like structure. Okay, it folds on itself and it forms a rod-like structure which is present like this. It folds on itself and it forms a rod-like structure. Okay, then this rod-like structure, it contains a very stable and self-cleaving and also self-ligating enzyme. It cleaves on itself and it ligates on itself, which is a self-cleaving and self-ligating ribozyme. Now this HDV RNA, it requires a host RNA polymerase II for replication. Okay, this host polymerase II will convert the genomic RNA to complementary antigenomic RNA, which is a plus strand. This antigenomic RNA will serve as a template for subsequent genomic RNA synthesis. Okay. So subsequent genomic RNA is synthesized by this complementary anti-genome RNA. So for this conversion we need host RNA polymerase 2, because this HDV does not have its own RNA polymerase 2. I'm really sorry. This HDV RNA, it has only one open reading frame, and it has a delta antigen, which is HD antigen. This HD antigen, it is a product of antigenomic strand RNA. Okay. Now this HD antigen, that is hepatitis D virus delta antigen, has two forms. One, it is small, and the other, it is large. The small one has 195 amino acid species, and it plays an important role by facilitating hepatitis D viral RNA replication. And this large one has 214 amino acid species, and this will suppress the hepatitis D viral replication. And this is mainly required for the assembly of antigen into the virions. The antigen for this assembly of antigen into virions, this large one is important. Now, what does this delta antigen do? This delta antigen, it binds to RNA polymerase 2 of the host, and this will stimulate transcription. So now the complete hepatitis D variants require the cooperative function of helper function of hepatitis B virus. Now, hepatitis D virus can infect with hepatitis B virus in majorly two ways. In the first way, hepatitis B virus and hepatitis D virus infect simultaneously. This is called as co-infection. Here, in an already hepatitis virus infected person, if hepatitis D virus enters, infects, then that is called as super infection. So there is co-infection and super infection. Now, the HDV virus infection, it is transmitted from a donor with, if the HDV infection is transmitted from the donor, if the donor think that it has group A or some subtype of HBs antigen, okay, if this has HDV virus, and then this HDV virus now transforms from this donor to other person who is a recipient of second group subtype of HBs antigen. So now once it has reached here, the HDV virus will assume the second subtype of HBs AG. It does not assume the first subtype. Okay, first subtype it will leave here itself, and it assumes has second subtype directly. So this is an important characteristic of hepatitis D viral antigen. Now, the hepatitis D viral antigen relies completely on hepatitis B viral virus, right? So because so the duration of HDV infection, it depends completely on duration of HBV infection. If HBV infection is present for longer time, then obviously HDV infection will also be present for longer time. Now, HDV antigen, it is expressed in hepatocyte nuclei and also and it can also be detected in serum. Okay. In acute infection, then first HDV increases and then anti-HDV of IgM and then anti-HDV of IgM class predominates and then it decreases, and then anti-HDV of IgG will persist for some time. Okay, this is HDV antigen. This is anti-IgM HDV. This is anti-HD IgG. Okay, so this is what is generally seen. This is in acute case. In chronic case of HDV, in that case you can see the HDV antigen is this HDV antigen is not detected in all the places. HDV antigen is not detected in liver, in serum. Remember that. I forgot to mention that. Okay, I'll draw this again because I have made a mistake. Okay. In acute and chronic. In acute form, if you see HBV infection, HDV, anti-HDV, then there is decreased IgM, and then if you wanted to know about IgG, like this. Here the IgG is also increased and IgM is also increased simultaneously. So this is the major thing. So first, if you see HDV, anti-HDV, this is IgG, IgM in acute phase. In chronic phase, anti-HDV, IgG, IgM. So this is about the hepatitis D viral infection. So let us learn the other virus, which is hepatitis C virus. So the hepatitis C virus, it is often called as non-A and non-B hepatitis. Okay, it is a linear, single-stranded, positive sense, 9600 nucleotide RNA virus. It belongs to the genus Hepatitis virus and family Flaviviridae. The HCV genome, it contains single, large, open reading frame, which is a gene, and this codes for virus polyprotein of 3000 amino acids, and this is cleaved to produce 10 viral proteins. Okay. Now the 5' end of genome has an untranslated 8 region adjacent to 4 structural proteins. So if this is the 5' end, it has an untranslated region and it has 4 structural proteins. So what are the 4 structural proteins? The 4 structural proteins include 4 structural proteins are nucleocapsid core, nucleocapsid core protein, which is called as C, 2 envelope glycoproteins. These envelope glycoproteins are called as E1 and E2, and there is a membrane protein which is P7. So the 5' untranslated region and the core gene are highly conserved among genotype. So these are not code, these don't code to many of these proteins, whereas the envelope glycoproteins which are there, these code for hypervariable region, which varies from isolate to isolate, and this allows the virus to evade the host immunological containment directed at accessible virus envelope proteins. So this will protect the virus against antibodies or host immunity. Okay, then the 3' end also includes, also it has untranslated region, and genes for 6 non-structural proteins. So here, if this is the 5' end, this is the 3' end, this has an untranslated region, and then it has genes for 6, 1, 2, 3, 4, 5 and 6 proteins, non-structural proteins, that include NS2, NS3, NS4A, NS4B, NS5A, NS5B. Okay, the 6 non-structural proteins. Now NS2 cysteine protease. If this is the gene and this is NS3 and this is NS2, then it leaves between NS3 from NS2. So this leads to, this is NS3, this is NS2. Okay, it leaves between NS3 and NS2. Okay, the next NS3-4A serine protease. It leaves all downstream proteins. from polyprotein. Okay. Other proteins which are involved are NS3 helicase, NS3 NS4 serine 4A serine protease, and NS5B RNA dependent RNA polymerase. HCV does not integrate into host genome. Now